hopefully one of the last Zoom meetings that we'll have to endure for a little while. I'll begin, of course, by acknowledging the traditional owners of the lands upon which we're all meeting or sitting. Here I'm in the Charles Perkins Centre today, and so I'd like to acknowledge and pay respects to the Gadigal people of the Eora Nation, elders past, present, emerging, and any Aboriginal people who are Torres Strait Islander people with us today. So this is the 10th year, the first decade that we have had this extraordinary summer scholarship program. The first cohort of 6 students joined in 2012-13, and since then we've had 120 undergraduate and postgraduate coursework university joining us for these summer scholarships. They're funded by a wonderful gift. It's the Nicholas Catchlove Early Career Research Development Fund, a fund that was established in memory of Dr. Barry Catchlove's son and Penelope Catchlove's brother, Nicholas. And we're so grateful for Barry and his wife, Louise Sylvan's ongoing support for our early career researchers. More generally, and particularly the opportunity this program provides to bring the brightest young students into the Charles Perkins Centre, and in many cases to retain them. We've had PhDs and postdocs and award-winning students emerge from this program and go on to careers elsewhere around the world. And it's been one of the, I would say, one of the most important wellsprings for the development of our talent within the Charles Perkins Centre. It's been a wonderful program. This year, we were incredibly fortunate to be able to augment the funding that came from the Catchloves for additional students. And that was thanks to commitments that came from supervisors, labs, partner organisations, which included Sydney Business School, the Menzies Centre for Health Policy, Sydney Local Health District, some of the medical research institutes, Heart Research Institute, Woolcock Institute of Medical Research as well, and others. And really what this showed us was 2 things. One, the quality of application. We had a large number of really outstanding applicants, and also the high regard in which these scholars are held across the Charles Perkins Centre. People were willing to commit their own funding to take advantage of the best of the summer scholar applicants. Now, it hasn't been easy. The students had to commence their projects between October and December last year and complete 8 weeks of research with their supervisors and lab colleagues. And of course, that wasn't made easy by Omicron and the whole pandemic. So I'd really like to acknowledge the additional challenges that students and their supervisors faced with the COVID restrictions, and just congratulate you all on being able to complete these projects. As is traditional, we hear from the scholars on today's event. We give them a rather short time to present the findings from their work, and an even shorter time for us to question them. And today we need to get through 22 presentations. And these students, you'll see, come from faculties spanning science, medicine and health, engineering, and arts and social sciences. So as is typical and indeed typifies the Charles Perkins Centre, we're working across disciplines, across faculties, and that's the hallmark of these scholarships. You'll find a link to the chat, and what we would like you to do is ask questions through the chat as our students are speaking. Each presentation will only last for 3 minutes, using 4 slides as a maximum, and be followed by a brief Q&A lasting at most 2 minutes. And what we'll do is allow you to join the discussion by adding questions in the chat box or unmuting yourself at the conclusion of each presentation. Normally, we would be slowed down by handing over a rather nice Charles Perkins Centre sheet, which gives you evidence of having been a scholar. We'll provide those subsequently, and we would normally take photographs as well, and we'll have to arrange that subsequently. So we'll have a slightly more streamlined process as we don't have to stop for those things. So there's going to be, you'll see the program in the chat that gives the full list of speakers. And I'll kick off with our first student presenter, Salil Barrett. So Salil, over to you.
All right, let's bring my screen up. All right, is that all clear?
That's great, thanks, Salil.
Okay, so I'm an arts student, but I also have a microbiology background. And my overall approach is looking at what we call the unit problem in microbial communities and how that connects with dysbiosis, which I'll talk about in a moment. Before I get into that, though, I want to say a quick thanks to my supervisors, Paul and Andy, for their advice and feedback over the course of this project, and as well to Dr. Dan Faith, who was kind of roped in a bit later on, but also gave some really useful insights that build into what we're sort of working on now. So starting off with this problem of dysbiosis, we can think about dysbioses as sort of disease-associated microbiome states. And these are sort of particular arrangements or systems of microbes in the body that are associated with a disease in a context that we can't quite think of as an infectious disease. They are often associated with diseases that are the focus of things at the Charles Perkins Centre, so obesity, diabetes, those kind of chronic health issues. And we can think of them also as a property of the entire host microbiome system. So not just of the microbial community, but also of the entire sort of networked interaction between the human and, its associated microbes. So dysbiosis research is typically conducted through genomic profiling. So that's looking at how the genetic content of the microbiome is understood. And then from there, you can infer the content of the microbiome and then possibly its function. Unfortunately, though, there are no demonstrated causal relationships between these dams and disease, at least not in human cases. So this is a big problem if we want to target interventions towards these dysbiosis-associated diseases. And in order to rectify this, we need to develop a framework to guide research to how we can actually examine and better characterize these causal pathways. So my research process was necessarily multi-interdisciplinary. I took my microbiology side of things, and this is through Andy's node at the center, and we're looking at key definitions, methodologies, and the principles behind microbial ecosystem sort of interactions. And then I coupled that with philosophical work, particularly from contemporary philosophy of causation, the philosophy of ecology, and that's sort of the conceptual framework for how we understand ecological processes, and also the concept of function, which was quite interesting, and I'll talk about that on my next slide. So function is a weird term, and it kind of gets thrown around a lot, and it tends to refer to different things. So we're drawing some from the philosophical literature, as well as the way it's used in more empirical work. We sort of came up with 5 key types of function that all refer to specific different kinds of processes in the microbiome. So you can see that in this table here. And then attached to those functions, we're putting forward these things called Emus, or ecologically meaningful units. I'll talk more about those in a sec. And these are the sort of the discrete things that we can use to describe a particular function. So depending on what function you're looking at, I think we need to have different ways of sort of cutting up the microbiome in order to get a better or more precise fix on what we want to be talking about. So defining Emus. This is the bulk of what I've been reading and writing about over the last 8 weeks. Emus are these question-dependent causes. I say question-dependent because if you think back to the function slide, what is meaningful for a particular feature or aspect of the microbiome will be dependent on the question that the researcher or the medical practitioner is trying to ask. So how we define or delineate these Emus will rely on multiple different factors. And we call these specificity, proportionality, and robustness. So specificity is about how, this is again drawing mainly from the philosophy of causation, is about how does the EMU, how fine-grained a control does the EMU have over a particular outcome. So this is a way of measuring sort of the precision of control that an intervention might have, for example. Proportionality, so it's about the resolution and how sort of precise and the scale of an explanation. Do we talk about whole microbiome systems or particular taxon within a microbiome? And then robustness, which is particularly important for these big public health issues, is does an EMU have a consistent effect across time and space? So does something that works in my system also work in yours? And this is a fundamental question if you're going to be guiding public health policy. Yeah, that was a very, very quick overview of our kind of early framework, and happy to take any questions that you might have.
Thank you very much, Salil. Now we have a question from David James. Can you describe one key experiment that has highlighted an important role for the microbiome in metabolic diseases?
Well, there have been lots, and the issue with saying putting down to an important role is the fact that we don't actually know, as far as my understanding goes, don't actually know what the role of the microbiome is, and that's why we need a causal framework to understand. We know there are associations between certain ratios or distributions of bacteria in the gut that might correlate to things like obesity, or the production of biomolecules by the content of the microbiome can connect with is associated again with certain changes in function, say in the epithelium of the gut, but that isn't causally demonstrated. So the question about the role is a bit tricky to answer without sort of making a whole bunch of presumptions. Yeah.
No, good answer, Salil. And one quick one. Jennifer Byrne has asked, what are your thoughts about integrating information between and across EMUs? And that seems to be a central question in relation to your project.
But between and across, I'm going to take that to mean sort of how do you compare different EMUs when you're in different functional levels? I think that's what the question's asking. Yeah. And again, that comes back to the idea of it being question dependent. And in some cases, a particular EMU will have meaning for an outcome that the investigator is looking at. In other contexts, it might not. So this is more about a process of trying to identify these things in order to guide research and guide interventions in the future, rather than saying this is how we should be thinking about the microbiome specifically.
Fantastic. Thank you, Salil. I think we probably now need to move on. And I can introduce our next speaker, Ruel Chua. Can we get your talk up?
Of course, yes. Hello.
Ah, there you go. I can see you. Brilliant.
Awesome. All right. So hello, my name's Ruel. I'm a second-year medical student. And for my project, I did on a literature review of the literature surrounding this topic of the mother's socioeconomic factors. So we would say rather demographic factors, and if those would have an impact on the acceptance of a COVID-19 vaccine for the young child. Yeah, so I had two main goals. The first is to obviously identify and synthesize. In terms of the literature itself, regarding the demographics of mothers and rather parents, and first of all, acceptance of the vaccine for their child, but also secondly, regarding the acceptance of the willingness to test and isolate when symptomatic of COVID-19. So the background, obviously everyone knows coronavirus is pretty bad, not something that you want to have around. And as a result of that, the Australian government's action plans to suppress and prevent the spread. Oh
dear. I think Ruel's frozen.
Will may have frozen.
Yeah.
Oh, it looks like he might have dropped out there, Steve. We'll give him, oh, he's joining again.
Back again. You dropped out then, Ruel, sorry. And you're on.
And I'm on mute as well.
You're on
All right.
mute.
All right, thank you for waiting. So yes, so recently the vaccine for children aged 5 to 11 was released just last month. So next up in line for vaccinations will be children 4 and under. So with our findings, we've found that female parents were more hesitant. So BAME populations in the UK were less willing. In Australia, there was another study that showed that participants that spoke a language other than English were also less willing to vaccinate. Low economic status and low education were potentially associated with hesitance, but other studies didn't find any association with these factors. In terms of age, one study interestingly identified that parents between the ages of 25 and 44 were more hesitant, and another study found that just younger parents were more hesitant. Again, these studies, there were other studies that found that age wasn't any indicator of hesitance or acceptance at all. With the second part of the review, there were only a few studies that asked about the willingness of mothers to isolate and test. But in these ones, they asked if they were willing to do so when either ill or having a confirmed infection, rather than being symptomatic, like what we wanted to investigate. In any case, there wasn't any significant links that were found. So yeah, in summary, Australia's approach to COVID-19 is to suppress its action and spread. And next on the agenda would be vaccinating children aged 4 and under. We found some factors that were identified to potentially have an indicator of a parent's hesitance to vaccinate their child, but others were contested as well. The literature describing the relationship between a mother's demographics and her willingness to test and isolate when symptomatic of COVID-19 should be expanded. We don't really know too much in that case. Last of all, I'd like to thank my supervisors, Professor Liming and Luis. I wouldn't be here without them and presenting in front of all of you. So yeah, thank you.
Thank you, Ruel. Terrific. We have a question. Bandana Saini asks, great topic, Ruel. Which countries were these studies you reviewed mainly reported from?
There were a bunch actually. It was all around the globe. There were some there were some that were from Turkey. There was one particularly from Australia, which I included, that had sort of reflected the main findings that we saw in other countries as well. But yeah, all around the world. Yeah.
Terrific. Oh, thank you. There's no other questions. So no, hang on. We have one, Davina Nair. How do you believe we can adapt policy or educational resources to assist with historical mistrust in marginalized communities? That's the big question.
Yeah, so I think, no worries, I see that. So I think this is a very difficult question to answer in the sense where, there's not one solution that can fit every community, every individual even has a very different experience with, the institutions or and the government, and as a result of that, obviously have that sort of sense of mistrust. I understand that. So I think to begin, I think a sense of empathy should be explored, I think, and just cooperation with the communities out there in the sense where, there's no one-size-fits-all education program or policy that mandates things, but rather work hand in hand with these communities to, pretty much create a more robust response to the pandemic.
Very wise. Yes. Thank you, Ruel.
Thank you very much.
So our next speaker is Sabah Fatia.
Thanks, Steve. I'll bring up her presentation.
Okay. Some of these are pre-recorded, by the way, and I haven't yet said thank you to Sarah and Nancy for helping all this work. I'll thank them fulsomely at the end as well, but Sarah is doing a brilliant job, as is Nancy. Okay.
Hi, my name is Saba, and I have been researching the impacts of intermittent fasting on islet cell function in healthy, unmodified adult mice. Intermittent fasting is an up and rising dieting technique that has been particularly effective in showing weight loss. There has also been research to show that weight loss can recover beta cell function, which leads us to contemplate as to whether an effective weight loss method such as intermittent fasting can also have beneficial effects on beta cell function and in particular insulin release. Ultimately, such research aims to aid the understanding and reversal of type 2 diabetes. In investigating this, we undertook an experiment where 8 healthy mice were equally divided into intermittent fasting diet and control groups. The fasting group were given food for 24 hours and they were followed by a similar period of food deprivation. At 1 week of this protocol, the mice were all included in a glucose tolerance test, and at 2 weeks, their pancreatic islet cells were harvested to undergo a glucose-stimulated insulin secretion assay. This graph shows our results from the oral glucose tolerance test, and we can see that the fasting mice exhibited significantly lower blood glucose 15 minutes after being given that amount of glucose orally. But both groups do eventually return to a similar and insignificantly different basal value at 90 minutes. This graph shows our results from the glucose-stimulated insulin secretion, and we can see no significant difference in insulin release at basal recordings, but a significantly lower insulin release in fasting mice upon glucose stimulation. Taking both of these results, we can hypothesize that in fasting mice, the skeletal muscle may be more insulin sensitive. So where to next? Given the results we've found, it would be favorable to run such an experiment again and evolve a muscle sample in the insulin secretion assay we perform, as well as to run the diet protocol for a longer duration and observe more mice to hopefully form a more distinguishing difference between the intermittent diet and control group. Thank you for listening.
Well, that was wonderful from Saba. Is Saba joining us for questions, Sarah? Because I know some of the recorded.
No, she's not able to, but I did see Pete Thorne on the line in case there was any chats for Peter.
I know, I think we'll
otherwise we'll move on, yeah.
And yeah, fantastic piece of work, really interesting. So thank you to Saba, and our next speaker is Koko Juan.
All right, let me share my screen nice and quickly. There we go. Yep, can everyone see the screen and hear me okay?
That's great, Koko, thank you.
Yeah, awesome. Okay, well, good morning, everyone. I'm Koko, and I'll be presenting my summer research on the role of macrophages in impaired wound healing in diabetes. I'm sure everyone here has heard of diabetes, but lesser known is a complication that occurs in up to 1 in 3 people with diabetes. These nasty foot ulcers you see here can arise from delayed skin wound healing, and they can lead to poor quality of life and even amputation. But what is it that actually impairs wound healing? So macrophages, which are a type of immune cell, play a key role. They're generally classified as M1 pro-inflammatory or M2 anti-inflammatory, and they help the wound to transition from an initial pro-inflammatory state to an anti-inflammatory pro-regenerative state. They also stimulate fibroblasts and keratinocytes, which are types of skin cells, to close and repair the skin. So fibroblasts produce collagen, which is a type of structural protein in skin, and also tropoelastin, which is a precursor to the protein elastin, which enhances wound healing. However, in chronic wounds, like in foot ulcers, the macrophages accumulate and there's uncontrolled excessive inflammation. How these M1 and M2 macrophage proportions change during wound healing and how diabetes could come in and affect the whole balance and disrupt healing is unclear. So that's why we aim to examine whether this imbalance contributes to delayed wound healing in diabetes using a cell culture model. We hypothesized that a high glucose environment with imbalanced macrophage profiles would impair wound closure, but using tropoelastin developed by the Weiss lab and restoring that M1-M2 balance would improve it. To test these hypotheses, we performed scratch wound assays, which involve growing cells in plastic wells and then making a scratch through them to simulate a wound. The wound was then imaged over 7 days using this IncuSite live cell imaging machine, and wound closure was digitally quantified. So firstly, we cultured either the fibroblasts or the keratinocytes alone or in a 1:3 ratio. And the wells were coated with collagen, tropoelastin, or no coating. They were given media with normal or high glucose concentrations to mimic normal or diabetic conditions. And we found that this 1:3 ratio, the collagen coating in green, and also, surprisingly, the high glucose conditions, which are in the dotted lines, had the fastest wound closure. And this could be because that the fibroblasts themselves produce extra collagen and tropoelastin, but alone you can see the fibroblasts can't achieve the full wound closure. And that just highlights how that 1:3 ratio is really important for the fibroblast keratinocyte crosstalk in helping wound healing. Then we grew monocytes, induced them into macrophages, and then into M1 and M2 macrophages, the pro and anti-inflammatory ones. And we collected the media they were cultured in and treated the cells with these in different ratios. So adding conditioned media from macrophages, especially M1, M2, and the combined ones, actually reduced the speed and the extent of the wound closure compared to the control. Also, the tropoelastin in orange and collagen in green had very similar trends, which reinforces our findings in another trial where collagen and tropoelastin both improved wound closure compared to the uncoated wells. So in summary, wound closure was optimal when the fibroblasts and keratinocytes were co-cultured in a 1:3 ratio, wells, and also when the wells were coated with tropoelastin or collagen compared to uncoated ones. But treatment with the macrophage conditioned media inhibited wound healing, and the pro-healing effect of high glucose should be further investigated. Further studies could use this model to investigate how tropoelastin or other novel biomaterials could modulate the behavior of macrophages in wound healing, which could help us develop new chronic wound treatments for many people with diabetes. Okay, thank you to my supervisors, Dr. Danjing Min and Professor Tony Weiss, and all these people for their training and support. Thanks for your attention, everyone, and I'm happy to take any questions.
Thank you, Koko. That was an extraordinary amount of work in 8 weeks. Well done. Do we have questions? I'm intrigued by that glucose result, Koko. What was high glucose? What sorts of concentrations are you talking about?
So high glucose was 25 millimolars of glucose in that cell culture media. The reason why I think the high glucose could have actually improved wound healing is that the skin cells, the fibroblasts and keratinocytes, when you're maintaining them between and before trials, the recommended concentration of glucose to maintain them in is actually also high glucose. So suddenly switching them to normal glucose may have actually impaired their wound healing. To improve this for future studies, I would think it would be a good idea for us to actually get keratinocytes and fibroblasts, extract them from people with diabetes, so use primary cell culture rather than these cell lines. That would probably avoid that problem.
Yeah, makes sense. Great. In the absence of any other questions, thank you very much, Koko. We will move on now to Salma Gabara is our next speaker. And it's pre-recorded.
Thanks, Dave.
Thanks, Sarah.
Hello, my name is Salma, and I'll be talking to you about my health decision-making tool that I've created for this project. So the ultimate objective of my project was to build an online platform that serves as a research instrument for examining how pre-diabetics make decisions about managing their health. So this involved extensive research into prediabetes, type 2 diabetes, and the many lifestyle components that are involved with reducing the risk of a pre-diabetic developing type 2 diabetes. And I've also used Qualtrics to build the online platform, and I've used many of its features as well as my own JavaScript code in order to enhance the functionality of my online platform. So the specific factors that I was measuring were the participants' preferences and priorities in terms of the lifestyle modifications that they would make to reduce their risk of developing type 2 diabetes, and the influence of evidence-based information on their decision-making. So while taking part in my experiment, the participants' goal is to create a lifestyle plan that reduces their risk of developing type 2 diabetes. And they do this in 3 simple steps. The first step involves creating the initial draft of their lifestyle plan. They do this by answering a set of questions based on how they prefer to include the important lifestyle components of physical activity, diet, and sleep into their plan. Here's a quick example of what one of the questions could look like. And my purpose of these set of questions is to get a sense or measure their preferences and priorities in terms of lifestyle modifications. The second step involves the participants to read and navigate the evidence-based information presented to them about the most effective lifestyle approaches a pre-diabetic could take for reducing their risk of developing type 2 diabetes. The third and final step allows the participants to review their draft lifestyle plan that they created in step 1 and edit their plan if they would like to do so. If they make any changes to their plan, it will prompt a question asking them for the reason why they changed their mind. By comparing their answers in step 1 with any changes they've made in step 3, I plan to analyze whether the evidence-based information that was presented to them had an influence on their decision-making. So what's next? I plan to conduct user studies for several reasons, and here are some of them. So to examine how the participants interpret and navigate the evidence-based information that is presented to them, to examine how they interpret the questions asking for their preferences, and to give insight into how they make their decisions or the reasons behind their decision-making. The user studies will use a think aloud methodology where they verbalize their thoughts throughout the entire study. The users will act as if they're making the decisions about what they should include within their lifestyle plan, which I've mentioned before. But what I would like to add to that is different users will see different versions of the platform to get a sense of how different ways of presenting information changes their thought processes. Thank you for listening to my presentation.
Well, thank you to Salma. Is Salma's not with us for questions either, Sarah? Okay.
No.
Terrific. So Micah and Michelle, Micah Goldwater in psychology and Michelle Koenig, who's our health economist, are the 2 supervisors. So questions could be directed afterwards through Salma to them as well. Thank you. So our next speaker is Harry Juan. And we'll hand over, he's here in person, isn't he? So we'll hand over to Harry.
Thank you, Stephen. Could everyone see my slide? Hi, so good morning, everyone. My name is Yan Song, and my project is solving the compatibility issue between Icg devices and electronic medical record systems within Aboriginal medical services. My supervisors are Dr. Josephine Gwen, Professor Ben Perframont, and Dr. Kelly Gwen. I'd like to begin by acknowledging the traditional owners of the land on which we meet today and the Gadigal people of the Eora Nation. I'd also like to pay my respects to the elders past and present and to the communities taking part in the research program. So firstly, the Icg device is also known as single lead electrocardiograph device. As you can see on the right-hand side, It is a handheld device connected to a mobile phone or tablet. Similar to the traditional 12-lead ECG, the iECG also detects atrial fibrillation, the most common type of heart arrhythmia in Australia. Early screening of AF is important for Aboriginal Australians because the prevalence is higher and present at a younger age than non-Indigenous population, as identified by the research team. The EASY project is led by my supervisors and aims at the early screening of AF for Indigenous Australians using iECG devices, and this project is about to be submitted for the MRFF grant. However, there were some compatibility issues between electronic medical record systems and iECG devices during the trial. This could be a barrier when promoting this program in Aboriginal communities. And my summer project was aiming at investigating these issues for the 4 different EMR systems. The method I used for this research was key informants interview. There were 16 key informants and stakeholders involved, including AMS managers, clinicians, software engineers, researchers, and IT officers. 10 questions were involved in the interview, 9 informants attended the interview, and 5 was consulted via email, and 2 informants didn't reply. A feasible solution was identified through content analysis of the key informants' interviews and emails. So the video playing on the right-hand side is a demonstration of the iECG device recording my heart rhythm on the mobile phone. After a 30-second recording, the patient ID number was put in and the PDF will be sent to the staff at the Aboriginal Medical Services. They will look up the patient ID number and upload the trace to their medical records. When data extraction is needed, clinical audit will be made using softwares like PenCat. The result will be for the patient and follow-up pathway commences. Although all key informants agreed that this pathway is feasible enough, we would like to make this process easier by making the entire upload process automated. And we are working with software companies to make it happen. The information from this project will contribute to the establishment of a co-designed AF screening and follow-up pathway with Aboriginal Medical Services. And lastly, I would like to thank CPC for this opportunity and all my supervisors for their support during the research. Thank you for your time.
Thank you, Harry. That was a fantastically important project. And so questions, do we have questions on chat, or would anybody like to unmute and ask a question? So you were saying, here we go. That was incredible, Yan Song from Camilla. So well done. That's I think we all agree with that. And Salil has asked, what would be the expense of this kind of technology?
Yes, so for the expense, so for example, we are participating with about like 10 Aboriginal Medical Services at this point. So the expense is going to be one device, like with the iECG device, plus an iPad for each Aboriginal Medical Services. That's going to be like about 1,000 Australian dollars. And we are applying for the MRFF fund from the Australian government. It's run by the NHMRC, and the grants will be at least probably like 200K if we succeed. And we think it's going to be like really sufficient for continuing this project. And it's going to be like a 2-year-long project at least to further implement like this AF screening project in the Aboriginal communities. So yeah, fingers crossed.
Yes, indeed. Fingers crossed on that.
Yeah, thank you.
The MRFF grant. Terrific.
Thank you.
Right, thank you, Yanson. And we'll move on now to the next pre-recorded talk, which is Serlin Lauren.
Thanks, Dave.
Hi everyone, my name is Serlin, and my summer project is on investigating iron levels in stiff heart failure following prophylactic NR supplementation. Heart failure with preserved ejection fraction, or HFpEF, is the increased non-compliance and stiffness of the heart. The risk factors and comorbidities include hypertension, obesity, diabetes, and iron deficiency, which we're interested in. Depletion of myocardial NAD has previously been linked to the pathogenesis of HFpEF, and supplementation of NR, an NAD precursor, has shown therapeutic potential to rescue HFpEF by replenishing depleted myocardial NAD. In an unpublished study by our lab, we observed that expression of several cardiac proteins involved in the regulation of labile iron pool and iron sulfocluster formation are dysregulated in mice HFpEF heart. These protein expressions were brought to normal physiological levels with the use of prophylactic NR. However, further study is required to see how these findings affect levels of intracellular iron in the heart and liver tissues. Based on this, the aim of our study is to investigate, if any, the presence of iron deficiency in murine HFpEF model, and also to investigate the differences in intracellular iron level following prophylactic NR supplementation. We hypothesized that levels of intracellular iron will be lower in mice with HFpEF, but normal in those supplemented with NR. Our experiment involves 3 groups, which is control mice, mice with HFpEF, and mice given prophylactic NR. Note that mice work was conducted separately and only frozen tissues were used for this paper. Frozen heart and liver tissues obtained from these mice were powdered and weighed into tubes in preparation for the iron assay. Total intracellular iron level, which are Fe2+ and Fe3+, was determined using an iron assay kit. Surprisingly, we found that the total iron in the liver is similar between control and HFpEF mice. This suggests that iron storage in the liver may not be affected in HFpEF. However, when compared to control, HFpEF mice showed a downward trend of total iron level in the heart. We think this might be caused by either iron transport problems from the liver to the heart, an uptake problem from the circulation to the heart, or an increase in iron demand somewhere else in the body. We also found significant reduction in total iron of the liver of prophylactic NR mice, which could be due to a transient benefit of using NR supplements by promoting iron uptake from the liver to other organs. However, we noticed that this increase in iron uptake to other organs is not reflected in the heart of prophylactic NR mice. This might be caused by impaired iron uptake of the heart, or the benefit of NR itself is too transient to see any significant increase in cardiac iron levels. Our study only measured total intracellular iron in liver and heart tissues. This is not enough to fully assess iron deficiency, as we did not assess other common measures of iron metabolism, such as ferritin. Also, because of limited tissues and iron assay kit availability, we were unable to fully investigate iron deficiency in HFpEF. So, to gain a better understanding of how iron deficiency is linked to HFpEF and whether the use of prophylactic NR may improve or even prevent iron deficiency in HFpEF, future studies should incorporate other measurements such as serum ferritin level.
Great. And Serlin's not with us, Sarah? No.
Sorry. Yes.
Excellent. So direct questions to Serlin and John O'Sullivan and Sean Lowell, the 2 supervisors, part of a big and really very exciting project. Okay, next speaker, Camilla Jones. And we've got Camilla.
Hello.
Hi.
I'm in person. Hello. Let me just share my screen. Okay. I'm not adept in, I don't want to share the wrong one. So let's try this. Okay, perfect. Okay, can you see just the screen, not my notes?
Yes, thanks, Camilla.
Okay, Good, thank you. So hi everyone, welcome to my presentation on safeguarding human health during hot weather and heat extremes in refugee camps. I'd like to acknowledge my supervisors and all of their contributions, Professor Ollie Jay from the University of Sydney, Professor Tony Cappan from Monash University, and our Medicine Sans Frontières conduit Associate Professor Lachlan McIver, who couldn't join us today as he is in Geneva, and this is not a good time zone for him. So our project was born from the work that Prof Jay and Prof Cappan had published in 2021 as part of the Lancet Heat and Health series, and was designed to expand their recommendations for maintaining core thermoregulatory temperature for refugees. So we focused on the locations of Tanzania and Lebanon as per the recommendations of MSF Geneva, as per their focus on planetary health. And what we did was we conducted a short narrative review of the peer-reviewed and grey literature over the first 6 weeks of the project, and spent the last 2 synthesizing our findings to formulate our recommendations. What we found was that of all the refugees in Lebanon and Tanzania, approximately 45% of them were children. And as research had previously shown, young children, specifically from 0 to 4 years or under 5, were particularly vulnerable to heat. So we chose to focus our recommendations on improving their health. So the main problem is that when children are unable to cool down through convection or radiation, so transferring heat from the body to outside of the body, or evaporation, sweating, this creates changes in a child's behavior. So this might be that they play more outside to escape the heat in their tents, which can reach 10 degrees higher than it can outside, even though it's still very, very hot. Drinking excess water, which is highly likely to be contaminated, and to play outside in stagnant or contaminated water. And overall, this can increase their prevalence of diarrheal diseases, so such as cholera and vector-borne diseases like malaria in Tanzania and leishmaniasis in Lebanon, which is a parasitic disease that can cause lesions and fever. What we recommended was collecting ambient temperature and relative humidity data at each refugee camp and incorporating them into the World Health Organization's early warning alert and response system. This data can then be paired with clinical presentation data in health clinics to track and predict the effects of health and associated disease outcomes. It can also be used to predict resourcing requirements, such as water purifying tablets for the use of safe self-dowsing, water drinking, and clothes wetting, so wearing wet clothes on the body to cool down. One of the most valuable tools for thermoregulatory cooling is to create opportunities for air circulation, so such as through the use of personal fans, as it improves evaporation and convection. In resource scarce settings, however, in lieu of personal fans, which can be used inappropriately as tender, we recommended using industrial fans in public shaded places. In early 2021, UNICEF had created a high-performance tent for multi-purpose use, which are accessible, so for persons with disabilities, have connected electricity and are insulated for optimized climate control. So we also recommend that the new thermal insulation materials used from the high performance tent, which at the moment are only in multi-purpose room tents, be used for domestic household use, or if that's not possible, that more multi-purpose rooms be built for recreational day and night use for more people. Finally, at an individual level, we recommend children engage in personal cooling activities such as self-dowsing, wearing cold, wet clothing, and drinking safe, clean water. And by making clean drinking water available and accessible and abundant during heat waves through heat resource forecasting, these children are less likely to end up playing, drinking, or engaging in contaminated or stagnant water, and therefore reducing diseases such as cholera, leishmaniasis, and malaria. Thank you.
Fantastic. Thank you, Camilla. So, open to questions. Do we have any in the chat? My I guess obvious question, Camilla, is how do these recommendations become implemented?
So the next stage would be to well, our next hope is that we will work with MSF, obviously Monash, UCID, to turn this into a policy brief with the engagement of the largest stakeholders in terms of UNICEF or UN donations, which is speaking with the United Nations, with USAID and the Central Emergency Relief Fund. Because there is such a huge gap in funding for refugee camps, we would need to discuss where that gap fits and how we can do that resource loading, and then obviously speak with the World Health Organization to see how we can implement those changes in the emergency response system.
Great. Well, thank you very much, Camilla. I think we have one point from Jane Gavin, perhaps an industry collaboration to supply the fans, question mark.
Yes. Absolutely. So there are a lot of organizations outside of the United Nations and their counterparts that do supply things like cold chain boxes and storage devices. Some of them actually do provide air conditioning units that can be used in medical facilities and in where MSF workers work. So we would look to, yes, do an industry collaboration with someone who would supply those fans and obviously work with the engineers to determine the best placement and how that would work.
Great, thank you very much. Well, thanks so much, Camilla. And
thank you.
To our next speaker, Adelia Key, and I think Adelia is with us.
Thanks, Steve. Yes, I am. Let me just share my screen. Okay. So, hi everyone. I just wanted to share what I did over my summer research in the Greg Brown Diabetes Lab. As I majored in pathology in my med-sci studies, I wanted to study immune cell populations that modulate inflammation, particularly in non-alcoholic steatohepatitis, also known as NASH. NASH is a severe manifestation of fatty liver, which is a complication seen in type 2 diabetes. And histological features often include intrahepatic lipid vesicles, immune cell infiltration, and collagen deposition. And the model that our lab used was effectively recapitulated these features. As you can see, there were changes between the normal healthy controls in E, and then when diabetes was induced in F, and when the mouse went through high fat feeding in G and H. So there are heaps of immune cells that are involved in inflammation based on their phenotypic or functional markers. And our lab showed previously in human blood that the number of pro-inflammatory CD68 macrophages increased when diabetes was induced, whereas the ones that express CD163 and are injury resolving decreased over time with diabetes. But what about the ones in situ in the mouse liver? And can these subsets be quantified? These questions informed my aim of examining immune cells that modulate inflammation in diabetes and NASH, and I focused on these markers and quantified them through flow cytometry. Most of my summer was spent on methodology though. For some context, I had chow controls and mice that were induced with diabetes, and their tissues were embedded in wax. So we tried different protocols, dewaxing with dishwashing liquid, with xylene. We tried different digestion concentrations to optimize the cell yield. And overall, after a few tries, we got some results. So these are just representative, and we followed a common gating strategy, firstly gating for CD45 and F480, and we labeled this group as the infiltrating macrophages. And then we separately investigated them for the activated phenotype, which is CD16 expressing, or the anti-inflammatory phenotype, which is CD163 expressing. And overall, the group with diabetes showed an increased proportion of pro-inflammatory CD16 cells, whereas they had a lower proportion of these anti-inflammatory reparative macrophages. But the differences weren't statistically significant, and this could be because of contamination, low cell counts, and cell yields. So I'm still working on optimizing the methodology because this is not where I want to stop. In the future, I'm hoping to quantify and also gather geospatial data about these immune populations in the liver. So instead of cell suspensions, I will be processing these tissues on slides to visualize them like how some of these researchers did it in their papers. And now I just wanted to give my special thanks to my supervisors, Steve, Helen, Danching, you are legends, and then my peers and the CPC for this opportunity. Thanks, guys.
Thank you, Adelia. Very good. Questions? Have we got any questions in the chat? In which case I have one. How did you induce diabetes in your mice? What was the treatment? Was it a high-fat diet?
So they were fed a chow-fed diet, but they were given a low dose of streptozotocin. So it's a glucosamine for 20 weeks. Yeah.
Right. Interesting.
Mm-hmm.
Do we have other questions for Adelia? No? Great. Well, thank you very much. We're moving now on to Annabelle Perframont, and we've got Annabelle in person, I think, haven't we?
Yes, that's me.
Brilliant.
Okay.
Off you go.
Perfect. Okay. Just checking everyone can see my screen. Okay. Perfect. My name's Annabelle, and I've had the privilege of having spent the last 8 weeks undertaking a summer scholarship at the David James Lab, in which I've been investigating a mitochondrial protein, MIPEP, and its connection to diabetes. So perhaps some of you may have heard the term diabetes before. In fact, many of you may know someone that's affected or be affected by diabetes. Particularly the ABS, Australian Bureau of Statistics, reports that 85 to 90% of cases in Australia are type 2. So although there are a few different types of diabetes, the scope of my project focuses primarily on type 2. So how does a healthy person compare to a type 2 diabetic? Well, when a healthy person eats a meal, their blood sugar levels increase. So if we think this is almost like their bloodstream and this is sugar. And this causes an increase in insulin, which is a hormone that acts like a key to open up muscle and fat cells, allowing this glucose to rush in and therefore reduce sugar in their blood. Unfortunately, for people with type 2 diabetes, the muscle and fat cells have trouble recognizing this key, insulin, and therefore sugar in their blood remains high. And this is known as insulin resistance. And together with this insulin resistance, in fat cells of type 2 diabetics, the mitochondria has commonly been shown to be less efficient at producing energy. And this is referred to as impaired mitochondrial respiration. And so given this association of the mitochondria in type 2 diabetes, it becomes plausible that perhaps some mitochondrial proteins may be in fact involved in diabetes. And one of these proteins that I investigated was MIPEP. So how do we land upon this particular mitochondrial protein? Well, the lab that I undertook my scholarship in used this machine called a mass spec and found that this protein, MIPEP, was consistently downregulated in multiple models of insulin resistance. And so what is MIPEP? Well, it's a mitochondrial protease, and so it's involved in the processing of mitochondrial proteins. It resides in the mitochondria, which is where cells produce most of their energy. And so thus, for my project, I hypothesized that a downregulation of MIPEP in fat cells, also known as adipocytes, would therefore decrease mitochondrial respiration and insulin sensitivity. So how did I set out to test this? Well, first, thanks to a collaborator, we downregulated MIPEP in these fat cells or adipocytes, and they used this gene editing technology called CRISPR-Cas9, which essentially cuts the MIPEP target DNA. And this was done at 3 different places along the MIPEP genome to essentially create these 3 different independent MIPEP knockdowns. So I termed them RNA guide 1, 2, and 3. And once I was given these cells from our collaborator, I learned to culture these pre-adipocytes called fibroblasts with the MIPEP knockdown and differentiate them into adipocytes using a cocktail of drugs. And so once we knew that this MIPEP was downregulated in these cells, I set out to test my hypothesis. So does a downregulation of MIPEP reduce mitochondrial respiration and insulin sensitivity? So the first thing I did was run what was called a seahorse experiment, which measures the oxygen consumption rate. And what we did observe was that in the MIPEP knockdowns, they had a reduced oxygen consumption rate, thus supporting my hypothesis that they have, reduced mitochondrial activity and respiration. And so the next step was to assess insulin sensitivity. And I did this by measuring insulin independent and dependent glucose uptake. And in the absence of insulin, you can see that the knockdowns showed comparable glucose uptake relative to the control. However, when stimulated with insulin, in fact, the knockdown cells actually remarkably increased glucose uptake, contradicting my hypothesis and actually showing that they were, in fact, more insulin sensitive. And so I guess at the end of the day, what I learned was that the beauty of having a hypothesis is that you don't always have to be right. You just have to get an answer. So thank you. And now I'm open to any questions.
Thank you, Annabelle. What a lovely story. You didn't just not get an answer, you got the opposite answer, and that's even more interesting.
Yes.
It could have been no different at all, and that would have been less interesting, but having the opposite is really fun, isn't it?
Mm-hmm.
Questions in the chat? Andy Holmes, is MIPEP downregulated consistently across tissue types?
Well, the proteomics data was in cell culture in adipocytes, but what I did find interesting was that the MIPEP was downregulated in the insulin-resistant models, and I was kind of speculating, why did we have this opposite effect where it was downregulated, but we had this insulin sensitivity where we had proteomic data and it was decreased in insulin-resistant models, but it was increased in, I guess, my experiments. And I think what the difference was in that these insulin-resistant models, they had chronic insulin, I guess, doses, whereas mine was very acute. It was for a 20-minute 1 nanomolar insulin stimulation. So no, this was just in cell culture and adipocytes and not in vivo, so in tissue. But perhaps that was the difference between these two differences in vitro, an acute versus a chronic dose of insulin, and perhaps even leaning towards maybe a compensatory effect once you get a chronic, insulin dose. But yes.
Thank you, Annabelle. And I'll leave you with a final comment from Paulo Ferreira. For the first time, I understood what insulin resistance is. Great explanation, Annabelle. So well done on that.
Thank you. Thank you.
Thank you very much. Now, Sarah, that's taken us to where we were supposed to have a break 8 minutes ago.
Yeah, I think we're a little behind, Steve. I had hoped to have a short intermission, but I think in the interest of time, we ought to press on just so that we can try and get to noon as on time as possible.
Fantastic. Well, it's so engaging that unless you need to leave the room briefly for a comfort stop, then let's press on. I think we've got the momentum going beautifully. Next speaker is Chen Liu. And Chen, are you here?
Yes, I am.
Fantastic. Welcome. Off you go.
Sorry, I'm just sharing the screen.
That's great. We can see it, Chen.
All right, thank you. Have you experienced full body soreness after staying up all night? Have you felt your back sore after a long weekend of binge watching on your bed? We now know that a bad night's sleep increases your pain perception, and being sedentary makes you more susceptible to low back pain. But what about having both? Had a terrible night's sleep, then made a wrong decision to lay in bed all day binge watching YouTube. Studies have shown that the combination of poor sleep and sedentary behavior is associated with an increased all-cause mortality rate. And me and my supervisors are interested in how they together influence low back pain outcomes. My study pulled data from a cohort study that was conducted to look at the relationship between physical activity and low back pain outcomes. We used activity limitations due to low back pain as the primary study outcome, which basically translates to level of disability. And for explanatory variables, we pull data on physical activity, sedentary behavior, and sleep quality. We put all these data into the magical statistical analysis to look at three things. Number one, the independent association between each explanatory variable and the study outcome. Number two, the joint association between physical activity and sleep on the study outcome. And number three, the interaction between physical activity and sleep on the study outcome. And here's our findings. First, poor sleep quality was significantly associated with higher activity limitations due to low back pain, which means if you had a bad night's sleep, you are more likely to experience disability due to low back pain. And secondly, low sedentary behavior combined with good sleep quality was significantly associated with lower activity limitations. In other words, if you are an active person and always sleep well at night, your chance to experience disability because of low back pain is low. And lastly, there appear to be no statistical interactions between physical activity and sleep, meaning in this case, 1 + 1 does equal to 2. The combined effects appear to be simply added up by the separate effects of sleep and physical activity. However, this relationship is complex, and there surely needs to be more analysis to test out different variables and more studies on the biological mechanisms behind. So what can you take out of my today's talk? Well, although we're still unsure to what extent sleep quality and physical activity interact and together influence low back pain, but by sleeping well and staying active, even if you do have low back pain, you are much less likely to experience disability. Thank you, and special thanks to my supervisors and collaborators.
Thank you, Chen. So we've got questions for you. So Salil Barrett has asked, does poor sleep act as a predictor for sedentary behavior or vice versa?
Thank you for the question, Sally. In terms of this, it's obviously not part of my study, but looking at the literature, I think there are studies looking at the relationship between sleep and sedentary behavior, because we know that one day only has 24 hours. And then the more sleep you have, for example, the less hours you have to engage in physical activity, and vice versa. But the relationship is still very complex, and there are researchers who are studying this, but I think there's no a certain answer in terms of one causes the other or vice versa. Thank you.
Thank you, Chen. Without further questions, we'll move on. So thank you very much. And our next speaker is Annie Shulin Ma. So over to you, Annie.
Perfect. Can everyone see the screen?
Yeah, good.
Great. So diet is a direct modifiable risk factor of all-cause mortality. We wanted to further understand this interaction. So using the NHANES dataset, this study aims to investigate food groups and all-cause mortality by exploring macronutrient compositions through nutritional geometry. This surface plot describes the survival scores of the NHANES dataset from 1999 till 2014, originally developed in Nick Como's PhD work. Following on his work, my summer project identifies the dietary patterns based on survival scores through nutritional geometry. Nutritional geometry is a modeling approach which allows you to map out macronutrients, carbohydrates, proteins, and fat onto a 3D plane, with fat increasing as you move towards the zeros of the X and the Y axis. Therefore, any point within this plot captures all three macronutrients collectively, which is what makes nutritional geometry such a powerful tool. The colors here represent survival score. The blue bullseye has the lowest survival score of less than 0.9, which translates into the highest risk of death. We've categorized this as group 1, which has the macronutrient breakdown of approximately 50% carbohydrate, 16% protein, and 34% fat. On the other hand, orange represents the lowest risk of death with the highest survival score of greater than 0.96. So using nutritional geometry, we identified the composition of their macronutrients, and these participants were divided into 3 groups, which all have relatively lower risk of death. These are Group 2, high carbohydrate, low protein, Group 3, high fat, low protein, and Group 4, high protein, low fat. 36,000 US adults aged above 20 were included in the study. From these people, 4,700 participants fulfilled the 4 macronutrient zones we previously identified. The average age was 50, and they were generally classified as overweight. These people consumed an average of 2,200 calories per day, and there was an even split between females to males, with the biggest sample size coming from group 1. Foods consumed by these participants were grouped into 9 USDA food categories shown here. There was a difference in the absolute energy intake between the groups. However, this presentation will only focus on the isocaloric comparisons between the groups. Therefore, this bar chart represents the proportion of total energy from each food group. The white arrows represent statistical significance compared to group 1. Across all groups, the biggest source of energy came from grain products, followed by meat, poultry, fish, and sugar, sweets, and beverages. We also applied separate models adjusted for lifestyle and socioeconomic factors, but we didn't see major differences from the base model shown here. If we look more closely, we found that compared to the high-risk group, the low-risk group generally ate less meat. Translating this into calories, the high-risk group would eat 150 grams of steak, while the low-risk group ate only 90. The low-risk group also ate more fruits and more vegetables. So what does this all mean? Well, if your New Year's resolution, like many of us, is to start a healthier diet, then you can simply begin with small changes, such as having another piece of fruit or share your steak while you help yourself to a few more bites of vegetables. Before I finish, I'd like to thank my amazing supervisors, Michael, Helen, and Nick, for their patience, guidance through through this journey. I've learned way more than I could ask for. And thanks for listening.
Thank you, Annie. Fantastic. Questions? We got questions for Annie? So I guess the hanging question for me is the differences in energy intake that you found between those groups, and wondered if you could comment on that, Annie.
Yes, sure. So I will share the screen quickly. So this pie chart shows the proportion of total energy. However, I did mention that there was an energy difference between the groups, and that was shown in this bar chart. So group 1 with the highest risk of death actually had the highest calorie intake, and the 3 groups, which all have lower risk of death, these all had lower calorie intake. So that was what we discovered from the absolute energy intake. However, for this presentation, I only focus on the proportion of total energy.
Interesting. That's certainly something to pick up on, isn't it? That's very interesting. Okay, Annie, thank you. I think we might move on to Alyssa Mansour, who's also in the room.
Yes, I'm here. All right, okay. Hi everyone, my name is Elisar. I'm a 4th year Bachelor of Pharmacy student. And this summer I had the chance to work on a project on sleep health management in residential aged care facilities, alongside my 2 supervisors, Professor Bendana Saini and Professor Christopher Gordon, that I would like to personally thank again for guiding me and teaching me so much throughout this project, and the CPC, of course, for this opportunity. So as you know, sleep is very important for the functioning of our brain, and we know that aging can affect it. Although our elderly patients are vulnerable in that they're at high risk of side effects and drug interactions, sleeping pills are very commonly used, and especially in nursing homes. So the aim of this study was to assess the current management plans and identify the gaps and barriers that prevent optimal treatment of sleep disturbances in nursing homes. So this study was qualitative in nature. We contacted potential participants, mainly nurses and pharmacists, and the data was collected from 17 semi-structured interviews that were then transcribed and analyzed into codes on in vivo. And the first questions that we asked were demographic in nature, and you can see the results here on the right. So most of our participants were in the 20 to 34 age bracket. They were mostly female, and they were all nurses. And the interesting yet concerning result that we got is that only 2 out of the 17 nurses actually had a history in sleep health training. We then proceeded to ask the participants other questions that you can see here on the left, and this yielded 3 main themes. So the first theme was the knowledge and observations of sleep health by the nurses, and the factors that affected the sleep of residents are listed here. And the most common one was the most common factor that was mentioned was the comorbidities, essentially meaning other medical conditions like pain, dementia, anxiety, and depression. And the second theme was about the management of sleep disturbances in nursing homes. And the nurses used lifestyle interventions, such as the ones that you can see on this slide. However, pharmacological methods were also very much prevalent. And you can see this quote here, where essentially the nurse says that he has never seen a doctor not prescribe a sleeping pill when they reported sleep disturbances in a resident. And on the bottom of the slide here, you can see the most commonly mentioned drugs. And although they were shown to be effective in some patients, the nurses also reported their ineffectiveness with long-term use. And furthermore, some side effects were observed, especially with the use of benzodiazepines. So temazepam is a benzodiazepine, and it was the most commonly used drug, and increased risk of falls, dizziness, and oversedation was reported. So the question is their use worth the risk? Well, the answer lies here in the third theme. Although the effectiveness of non-pharmacological treatments was known, there were many factors that affected optimal management in nursing homes, such as the ones that you can see on the slides. So what does this mean? We need to develop evidence-based guidelines for sleep management in nursing homes and involve other allied health professionals, such as pharmacists, to intervene in drug protocols and control their use. Drugs should not be used as an easy solution, as it was seen in this study. Thank you for listening.
Wonderful. Thank you, Alyssa. That's really important. Questions? Do we have questions for Alyssa? From Salil, oh no, the other way, that was another sleep-related question from earlier. So the question about if patients are immediately or residents are immediately being put onto drugs, what is the incidence? Do we know of sleep apnea? And going back to those earlier talks, is there any program for identifying, diagnosing, treating sleep apnea in residential homes?
Yeah, so most of the sleep disturbances that were reported by the nurses, it was mostly insomnia. So that was the first, the most common one. And then some nurses also reported sleep apnea and then restless leg syndrome. So this was mostly based off insomnia, not really the sleep apnea, because with sleep apnea, the most common treatment that would be used would be like the CPAP machine. So this was mostly related to the insomnia, which was the most prevalent issue, the sleep disturbance. Yeah.
And Anne Elias asks, other patients ask whether they want to take sleeping pills.
Well, interestingly enough, a lot of the time the nurses actually said that patients would ask for the pill even when it would not be working. So it was in both ways. Sometimes, the doctors would prescribe it as an easy solution, but also we have to take into consideration the factors that are mentioned here in theme 3, which is, for example, a lot of the nurses struggled with, staffing ratios where they didn't have time to actually go through non-pharmacological treatments. We didn't specifically ask about if the residents wanted the sleeping pills, but a lot of the time the nurses did say that the participants asked for it as well.
Right, and one last question from Helen Parker. Great work, Alyssa. I'm interested to hear how this work goes with developing and supporting a culture of safe stroke sensible deprescribing, or at least not overprescribing in RACFs. The other, oh goodness, it's a very long question. The other non-pharmacological methods you noted are varied and present a lot of opportunities. Yeah, sorry.
Yeah, definitely. And these are great points there. And this is what we're aiming to go for, looking at deprescribing guidelines, which can also be put into place by involving other allied health professionals, such as pharmacists, and definitely looking like at specific guidelines and deprescribing guidelines. So that's a really good point raised there, and I definitely agree.
Great, thanks, Alyssa. Terrific. We'd best move on. And our next speaker is Alexandra Mcalpin. And Alexandra is with us.
Yeah, I'm just going to share my screen.
Thank you.
Okay, let me know if you can see that.
That's great, thanks.
Awesome. So my project, I explored the people's motivations to get involved in citizen science projects. And before I begin, I just want to thank my supervisors, Dr. Samantha Robotham and Professor Nick Enfield. So citizen science, where members of the public collaborate on scientific projects, has been identified as an effective tool to engage individuals and their communities in health research and interventions. And whilst the potential benefits of citizen science are increasingly recognised, researchers and practitioners are still looking to better understand how they can engage members of the public in their research. And stakeholders point specifically to a lack of understanding about what motivates people to get involved and stay engaged in citizen science. Social movement theory explores the development and outcomes of social movements, including the reasons why people participate in voluntary collective action. And in this project, I conducted a review of contemporary social movement theory literature and explored its relevance to citizen science participation with an interpretive analysis of 2 health-related citizen science case studies. And what I found were a range of helpful concepts that might help citizen science practitioners engage, attract, and inspire meaningful participation in their projects. So our first important lesson is that motivation is not static. People are motivated by a host of competing goals, which can include their own personal objectives like income or skill development, collective objectives like improving community health outcomes, as well as more emotional, relational objectives like pride, recognition, and belonging. What's more, these objectives change over time, and motivation has a direct link with both the expected outcomes as well as the everyday experience of participating. 2 pivotal moments of motivational shift have been identified in both social movement and citizen science projects, which are the initial decision to get involved and the decision to continue engaging. And this means citizen science practitioners need to design projects that speak to both long-term goals as well as short-term satisfactions. The next finding illuminated some of the more intrinsic or social motivations that get people to participate. And my review identified 4 prominent drivers of social mobilization, including the need to feel connection and belonging, agency and empowerment, pride and recognition, and pleasure and satisfaction. And these 4 elements have been well documented across various social movements. And interestingly, they also emerged as significant factors in our selected health-related citizen science case studies, which suggests that this framework might translate to other citizen science projects. So what this means for citizen science organizers is when recruiting participants, tap into existing social networks and frame project goals around the values and aspirations of the groups you're targeting in language that resonates with them. Create opportunities for informal interaction, including through the use of online forums, which can help create a sense of belonging to the project community and enhance social support within the group. Offering regular, meaningful feedback on project outcomes can also help participants feel acknowledged and empowered, knowing their efforts have made a difference. and making sure project tasks are enjoyable, or at the very least, easy to understand and perform. And again, opportunities for social interaction and meaningful feedback can also sustain enthusiasm in the short term, even when project tasks are challenging. Thank you.
Thank you very much, Ali. That is, it's really interesting to see the theory underpinning, particularly the citizen science project node and all the work that it's doing across a whole realm of different areas. So have we got any questions online? I was just going to ask you, do you have a specific example where all of those recommendations may have been followed? They perhaps followed subconsciously in some cases. Is there any way of seeing whether more successful engagement in citizen science has followed those particular recommendations compared with ones that perhaps weren't sustained?
Yeah, I think citizen science is kind of like quite a new perspective within public health research and interventions. So I think this is definitely kind of offering some recommendations to an emerging field. Even finding the 2 case studies was a bit of a challenge in itself. But it was interesting to me that even with the 2 that we selected, whether the researchers kind of went out looking for those factors, the citizens themselves really came forward, like that they were the messages that were kind of coming forward from the citizens themselves. And you know, for all intents and purposes, both case studies did seem to be relatively successful in attracting and recruiting a sufficient amount of participation. So that speaks well.
Yeah, translating into public health from, you know, there are clearly interest groups who care about frogs or whatever else it is that citizen science, particularly in environmental sciences, has been very successful in. You've got those passionate groups who care about frogs or turtles or lizards or whatever it happens to be. It's slightly more challenging, I guess, in public health. But yeah, fabulous. Thank you. All right. Next speaker is Layla Mihaj, and Layla's with us too, I think. So over to you.
Thank you, Steve. I'll just share my screen. Okay. Awesome. Can everyone see this? Cool. Hi, everyone. Today I'll be presenting my research project, which focused on establishing the role of fasting in the health benefits gained from calorie restriction. So calorie restriction is defined as the reduction in nutrient intake without causing malnutrition. It has long been reported that calorie restriction can have extensive health benefits and increase lifespan in multiple organisms, making it a promising first-line defense in the treatment of aging and cardiometabolic disease. In the past, it was widely thought that these beneficial health effects were the result of reduced calorie intake alone. However, more recently, it has been recognized that typical calorie restriction treatments also include a prolonged dietary fast. Therefore, looking at how fasting contributes to the health benefits gained from calorie restriction is an area of research interest. In our lab, a feeding regime has been developed, which allows us to study the effect of fasting on calorie-restricted mice using 3 diet groups. We have our control with unlimited feeding access, and then 2 CR or calorie-restricted mice groups, which are both given 20% less calories than the control. Our CR fasting mice, however, are only given their food once per day, while our CR unlimited mice have their diet diluted such that they have unlimited feeding throughout the day, but consume the same amount of calories as a CR fasting mice, which we can see here in this graph. In our published data from our lab, we noticed that there was a significant difference in liver triglyceride levels between the 2 calorie-restricted diets. My objective was to look further into the gene expression underlying this difference to understand how fasting may be affecting calorie restriction. For my analysis, I used 54 mice from the 3 diets. The mice had been on their diets for 18 months when they were culled at 21 months of age for analysis. First, RNA was extracted from powdered mice liver and it was quantified to be used in cDNA synthesis. Real-time PCR reactions were then performed, and these were for our target genes, which in this case were ACC and FAS. And these genes were chosen because they're both associated with de novo lipogenesis, which is the process by which the liver makes triglycerides. And then these were the results of our study. And while it looks like there is a trend, no statistically significant differences were found between gene expression and diet type. Therefore, our current results show that the differences observed in liver triglycerides between the calorie-restricted diets cannot be attributed to the impact of fasting on the FAS and ACC genes. It is interesting to note, however, that these mice were quite old at 21 months, thus may have climatized to the diet and therefore displayed dampened responses. Hence, future studies will be undertaken to analyse the mice at earlier time points. Also, these cohorts contained both male and female mice. Therefore, further statistical analysis can be undertaken on these current results to see if there are any significant sex-specific effects of fasting. And I'd like to say thank you to my supervisors and those at the Simpson Lab for supporting me in this research project. Thank you very much.
Wonderful. Thank you, Layla.
No problem.
Do we have questions? Here we go. Michael Skilton. Is the null result due to sample size stroke insufficient power, do you think?
Yeah, so we thought that that could potentially be one reason why we did get the non-significant result, because we had around 18 to 17 mice in each group. So it is potential that it was a small sample size. Yeah, other than that, some of the other things that we thought it might be is that these mice were quite old at 21 months. We also had a 3-month, I think, and a 6-month cohort. We did the 21 months analysis first for my project because we thought perhaps they would show the biggest response. But now that we've thought about it, we might think maybe they got used to the diet, and so maybe the response that we were looking for might be more evident in the earlier time points. So yeah, there are a few reasons. We're not too sure, but hopefully it will become clear with some future research.
Thanks, Layla. We've got another question from Helen. Helen's questions are always very long, so
that's all good.
You mentioned the mice have become acclimatized to the diet. Does this mean that the different types of fasting might be more of an intermittent treatment option as opposed to a long-term dietary solution? Should this be able to be applied to humans?
Yeah, so definitely the climatization that we mentioned, that's kind of just hypothetical. We're not 100% sure yet without analysing those earlier time points. It's kind of what we're hypothesizing. But if that was the case, Helen, you'd definitely be right in that we'd be looking at kind of something more intermittent, perhaps, when it becomes translatable for human treatments.
Fantastic. Thank you. Thanks very much, Leila.
Thank you.
Then we will move on to Davina Nair.
Perfect. I'm just going to share my screen now.
Thanks, Davina.
Let me start by asking, is our everyday neighborhood environment a neutral space? And if it is not neutral, how does the environment determine individual behavior? My research was centered around moving beyond the exploration of individual determinants of obesity, instead to understand the wider macro and contextual factors which influence this relationship. Specifically, I examined how urban environments, through their construction, can facilitate obesity-promoting behavior, which was linked to the broader socioeconomic dynamics within these spaces. Broadening this conceptual agenda, I was able to include disciplinary knowledge often neglected in this line of research. Originating from an art history disciplinary background, I was able to place my research in the nexus between arts and science. Grounded in this disciplinary perspective, I centered my research around the conceptual theory of psychogeography, which examines how the constructed nature of spaces has direct behavioral impact on individuals. Operating in this interdisciplinary space allowed my research to adopt an experimental research design. I incorporated methodological techniques from art history and visual arts and merged them with previously established scientific forms of inquiry. In my research, I examined 5 neighbourhood settings in the Greater Sydney region, in which for each of these areas, I used a combination of walking and driving to observe the constructed nature of these spaces. This type of fieldwork was inspired by the art group The Situationists International. To illuminate my findings empirically, I recorded two types of observational data: visual data through the use of photography and textual data through recording a list of all the retail food options within a neighborhood setting. From this data, I constructed a retail food environment index, which measures the density and quality of food choices within an environment. The research found that there was a positive relationship between the structure of a neighborhood food environment and the rate of obesity within a given area, which aligned with the principle of psychogeography and the impetus for the research in highlighting the contextual factors which influence individuals' behavioral choices. Further, when including the additional variable of socioeconomic status of a neighborhood, this trend ran parallel with the rise in both obesity and RFEI score, in which the research found that fast food restaurants clustered within lower socioeconomic areas. This is clearly illustrated in the visual imagery, which aimed to capture how these environments were structured. This emerging research ultimately points to the multitude of potential avenues for future research. For example, I originally aimed to use drone imagery to map the topography of the environment, which highlights how new technologies can be garnered in experimental ways to assist research. Further, the research is currently focused on the exterior and external environment. However, future research can instead focus on the construction of the interior landscape within food outlets and how this construction affects individual choice, which can utilize theoretical principles such as semiotics. Ultimately, the research highlights how there is a breadth of untapped knowledge from disciplines generally excluded from this line of inquiry that can prove fruitful in tackling these multi-causal and multi-dimensional global challenges. Finally, I would like to extend my gratitude to the Charles Perkins Center for giving me this opportunity to embark on this learning journey. And additionally, I would also like to thank my two supervisors, Anne and Jane, for allowing me to express my ideas freely, but providing me with invaluable guidance and for being so generous in sharing both their time and knowledge. Thank you.
Thank you, Davina. That was wonderful. Now, if we haven't got any other questions, yes, we do, because I've got a question. Mark Lawrence, would it be possible to use Google Street View imagery to map this on a much wider scale?
Yeah, absolutely. That was something I was looking into. But for me, coming from that visual arts background, I guess I wanted to be responsible for those images. And I think as utilizing kind of looking at that idea as a practicing artist, I guess Google Maps, it's taken by someone else. So that's why the drone imagery kind of came into fruition, which I did start to do, but unfortunately due to requiring permits and particularly the areas I was working in, I just wasn't able to do it in such a short space. But there definitely is potential to kind of use these sort of new technologies and use that in an artistic fashion, but then to kind of adopt and apply this to more scientific lines of inquiry.
I really love the idea of doing it across scales. So you're talking about applying the same approach to looking at, for example, the way in which different options are laid out within a supermarket or within a store, and scaling that up to neighborhood and landscape scale. That's really exciting.
Yeah, no, exactly. And I think it's about this idea that when you come from this sort of art history perspective, I look at the way kind of areas are constructed. And in doing so, you can kind of apply these to these varying scales. So kind of introducing and entering, say, a supermarket environment, the way like product placement is placed at this eye level, it's all to do with the gaze of the individual and kind of utilizing I guess that art history perspective of gaze and looking at it critically. You know, there's so much potential in sort of this sort of art research. Yeah.
Well, you've got lots of questions, actually. Anne Elias says, great ideas about visual images. Would you consider collecting data from interviews? That's one question. And I'll read the second one as well from Takwa Tisawi, which is, I'm assuming that taking photos yourself gives you insight into what it would feel like to be a resident yourself, and therefore the thought process of making decisions in these food environments.
Yeah, I guess so two of that. I'll respond to the second question first. I think definitely going into the environments, which what I did was I walked all these suburbs and I drove through them. And that really gave me the sense of the way these environments were kind of structured. And I guess there is potential, you know, the photographs very much capture that, but even using like a GoPro as a moving image as you walk to kind of capture that environment, I think is, you know, there's a lot of potential in that area. And then I guess also the second question in relation to collecting data from interviews, I think is a great way to actually get a sense as, yes, can be a researcher and come into the environments, but the environments can't just speak for the way people interact in that. And that's why having that personal touch through collecting data from residents is probably a very essential way, because there's the other thing is, you know, I'm looking at neighborhood environments, but people get their food from, like, you know, they might be traveling out an hour and a half to go to their workplaces. So actually understanding individuals and the way and where they're making these decisions is really important. So collecting data through interviews is a great line.
Fantastic. Well, we'd best draw it to a close. Jane Gavin says, great work, Davina, sharing this as a case study idea with urban planning colleagues and students at UCID could be a future avenue. And I think that's a really good idea. So thank you. Moving on then to a pre-record from Caroline's Song. So over to you, Sarah.
Hello everyone, my name is Caroline and my project is bioengineering tools to investigate neutrophil mechanobiology in thromboinflammation. So blood clotting with inflammation, therefore thromboinflammation, is well recognized in the context of sepsis, ischemia reperfusion injury, and the COVID-19 pandemic, and have led to severe symptoms and death. Therefore, elucidating the synergy between thrombosis and inflammation is crucial to develop anticoagulation therapies. So thromboinflammation impairs the endothelium's anti-inflammatory and antithrombotic functions, resulting in platelet and neutrophil activation, unleashing the complement system and coagulation cascade. So specifically, the P-selectin are adherent activated platelets induce neutrophil tethering mediated by its interaction with P-selectin glycoprotein ligand 1, PSGL1. So my project aims to investigate compression-dependent neutrophil adhesion to understand the impact of collision on neutrophil adhesion strengthening. So in order to simulate the blood vessel microenvironment, I used this microfluidic device that is as big as a thumb. The sample goes into the channel from the inlet and exits via the outlet. The channel is coated with P-selectin. Then washed human neutrophils is perfused through the channel at a typical venous wall shear rate, which is 150 /s, which simulating neutrophil rolling on activated platelet P-selectin. Then I tested neutrophil rolling under different conditions. Firstly, biomechanical. So when increasing the shear rate to 1000 /s, which represents high arterial shear rate, it was evident that there was no rolling neutrophils. Secondly, sample with biochemical treatments, including EDTA that chelates calcium, PL1, an anti-PSGL1 antibody, and G1, an anti-P-selectin antibody, were perfused through the channel under 150 /s. And as expected, there was no neutrophil rolling along the channel wall. So in conclusion, the P-selectin dependent neutrophil rolling only occurred under low shear. And EDTA, PL1, and G1 abolished neutrophil rolling under low shear by disrupting the formation of P-selectin and PSGL1 bond. Then taking from here, I've been using a cutting-edge bioengineering technique, biomembrane force probe, to investigate single molecule interaction under varied mechanical force. And in addition, both techniques can be engineered into a high throughput platform for anticoagulation drug discovery for thromboinflammation diseases. And I would like to acknowledge and thank Professor Sean Jackson and the Thrombosis Research Group at the Heart Research Institute and Dr. Arnold Zhu and the Zhu Lab for the guidance and support. Thank you.
Terrific.
We should have Caroline online.
Caroline's online. can you hear us, Caroline? Are you there?
Yes, I can.
Oh, fabulous. Well, that was a beautiful talk. Thank you. So we'll open to questions. Are there any questions on the chat?
Not yet.
So Caroline, I guess the next question is, what will you do with this next? Are you continuing this research?
Yeah, definitely. Because as I mentioned in the presentation, so the next step will be using the biomembrane force probe to actually investigate the single molecule interaction. And that would be basically my honors thesis. So if you're interested, I will invite you to my honors thesis presentation.
Fabulous. Well, we can't wait for that. So thank you very much, Caroline. I think we're going to move on to Natalie Smith, who's our next speaker.
Hi.
Hi, Natalie. Over to you.
Perfect. I'll just get started then. So I'm Natalie, and today I'm going to be talking a bit about my summer research project. So coronary artery disease is a significant cause of death around the world, which is why it's important to continue research into novel treatment strategies. And a growing body of evidence has demonstrated the role of the immune system in its pathology. So clinical interest in immune targeting therapies for coronary artery disease, or CAD, is growing. But how exactly can we study the immune system in the context of CAD? Well, this is where cytometry by time of flight comes into the picture. So an easily accessible snapshot of the immune system can be received by studying these PBMCs. And these are peripheral blood mononuclear cells. So they're the white blood cells in circulation that are collected just by a simple blood test. By phenotyping the cells in peripheral circulation, dynamic changes that might directly or indirectly be involved in the development of CAD can be detectable. And this is referred to as immunophenotyping. So cytometry by time of flight is an emerging technique for high dimensional immunophenotyping. So it can measure over 40 parameters on the single cell level, allowing a suite of cellular processes to be resolved in many cells simultaneously. So to give a bit of a brief overview of how this works, cells are tagged with antibodies that are specific to proteins of interest on the cell. And these antibodies are bound to a heavy metal isotope, which is like a tag. So when we mix the cells and the antibodies together, the antibodies bind to proteins of interest on the cell. And then we run them on the mass cytometer, and through a couple of pretty complex processes, we receive what's the iron mass profiles for the cells. And this essentially tells us which proteins are expressed on which cells. So pilot data from our collaborators demonstrated that these peripheral cells from coronary artery disease patients have high levels of ASGPR1. And this is a transmembrane protein that plays a critical role in serum glycoprotein homeostasis. So to bring it all together, this summer, I aim to examine the expression of ASGPR1 in these PBMCs from 14 people who had suffered a recent heart attack. So first we did a bit of a little pilot test with 5 samples to check the performance of this new anti-ASGPR1 antibody that we just purchased. And results from this test revealed dynamic changes in certain immune cell populations between the diseased people and the healthy control. However, when we went to examine ASGPR1, we detected virtually no signal in any cells, and this is far from what we'd expected. So this kind of hinted that there might be a problem with the antibody that we'd received from the manufacturer. So after receiving this unexpected result, my project took a bit of a pivot as we had to further assess this antibody. And after a series of tests, including some more cytov and flow cytometry, we actually determined that we had received a defective antibody from the manufacturer, and a replacement antibody has been ordered. So follow-up analyses will be performed to immunophenotype those PBMCs and define ASGPR1 expression as originally planned. But despite this deviation from my original plan, this project offered a really valuable experience in experimental design, use of controls, and troubleshooting, as well as an experience of the bumps that can come up in real science. So thank you to my supervisors for supporting me this summer and always encouraging me to learn.
Thank you, Natalie. They are incredibly important lessons, and you won't just learn them once in a career in science. That's one thing, but no, brilliant. You will understand CyTOF and all its intricacies better from that experience.
Yeah, exactly.
Yeah, oh, that's great. Very good. Have we any questions for Natalie? I can't see any on the chat, and so in which case, well done, and we'll press on, Natalie, if we may. So our next speaker is Ethan Italiano. And
yeah, I'm here
with us.
Just going to share my screen. Okay, can you see my screen?
Yep, great.
Awesome. So good morning, everyone. My name's Ethan, and I will be presenting my project on new therapeutic strategies to reduce microvascular obstruction, which is especially relevant to the current COVID-19 pandemic. All right, so we know there is quite a range of clinical manifestations associated with COVID infection, ranging from asymptomatic to mild symptoms to some really significant manifestations such as multi-organ dysfunction. Now, such dysfunction is attributed to endothelial injury, resulting in the development of a microangiopathy, which in turn leads to organ ischemia and enables the formation of microthrombi to obstruct vessels. So that is our microvascular obstruction. And conventionally, this microthrombi is associated with fibrin and platelet components. So this is where it gets a little interesting. Upon analysis of COVID-19 samples, the deposition of an acellular material was observed that is distinct from the conventional components. So of course, we aim to investigate this material, but before we can even approach this, we first need to showcase what normal fibrin looks like. So in order to do this, we need a special technique to allow us to observe fibrin, which is known as immunohistochemistry, or IHC. And this technique must be optimized first by testing a few variables within the protocol and comparing the results. So here we have two types of epitope retrieval, which is a process required to observe fibrin. The top row is a heat-based method and the bottom is an enzyme-based method. And this first column of images is just showing that we have vessels with fibrin as a positive control. However, when we turn on the fluorescent channel, we can see that the enzyme-based method has failed to present any signal. So once merged and optimized, we can clearly see the difference in fibrin staining, allowing us to implement the heat-based method as part of the main protocol. Now, once all of these variables have been tested and compared, we can then apply our optimized protocol and collect some usable data. For example, this tile scan of the fibrin staining in lung tissue. So this tile scan can guide us when taking higher magnification images like these on the right. Now in these images, we can actually observe red cells within a vessel and see that fibrin is indeed accumulating intravascularly within the vessel. Now, when we compare this to the clinical pathology gold standard, we can see our result has closely replicated normal fibrin staining, suggesting that our protocol has been successful. So what is the next step from here? So as we have now shown normal fibrin staining, we would need to repeat this for platelets, generating the full staining profile of microthrombi. We can then investigate the acellular material seen in COVID samples, which hypothetically should look vastly different, and that would confirm the absence of fibrin and platelets. Ultimately, after these confirmation steps are complete, we can then test our potential targets until we find out what this material is made of. This will provide us with a better understanding of the pathophysiology of COVID, allowing us to implement therapeutics in the future to reduce microvascular obstruction. So finally, I would just like to thank my supervisory team and members of the lab that made this project possible. Thank you.
Thank you, Ethan. Very impressive. So do we have questions on the chat? The big question, obviously, is I hadn't realized that there's an unknown material rather than the sort of expected inclusions that you would get in such microthrombi. So when was that discovered, Ethan? That's really important, isn't it? It's like novel biology.
Yes, so it is quite novel, and there haven't been any papers published on it yet. It's mostly just diagnostic H&E images going to pathologists, and pathologists looking at it and going, I see the fibrin, I see the platelets, but what's this material here? And nobody can really answer what the material they're seeing, like what are these like intravascular occlusions? So obviously before you can even investigate, you need to, exclude everything first, and then you can start doing your investigative studies and like looking into it. But yeah, very, very recent and very novel stuff.
Very exciting. So you have a range of comments and questions. I'll quickly read them out.
Yeah.
Takwa says, does this have the potential to help patients experiencing long COVID symptoms? Jane says, well done on your graphics and the flow of your argument, Ethan. Absolutely agree. Takwa also says, and thank you for the clear explanation. And Mark Lawrence asks, do you think the acellular material also comes from platelets? So maybe start with the last one.
Yeah. So from platelets, I haven't actually gotten the chance to test platelets. So I would need to do an optimized immunohistochemistry protocol for the platelets and then show that COVID samples stain negatively for platelets, so negative immunoreactivity. That would confirm that it does not contain platelets. Ultimately, however, it's more likely that it is a combination of a few different materials, just not in the same makeup that we see microthrombi in. So there might be a bit of neutrophil extracellular traps. There might be a tiny bit of fibrin platelets. But overall, the major component could be something entirely else, possibly lyse red cells, for example, which would be quite novel. And as for the long COVID question, if this could reduce the possibility of long COVID, I honestly am not sure. This is more aimed at therapeutics for multi-organ dysfunction, which is one of your primary manifestations. So long COVID will definitely need more research.
Right. Well, thank you. Excellent answers to some really good questions. So thank you. Thank you, Ethan. Next speaker is Daniela James New, and Daniela is with us.
Yes. Hi, I'll just share my screen.
Great.
Can you see that okay?
Yes, all good, thanks.
Okay, wonderful. All right. Okay, so my name is Daniela, and I wanted to start by thanking Dr. Giselle Yeo and Johnny Wong for the support and amazing opportunity to work with them over the summer. I've been helping work on a 3-dimensional expansion of mesenchymal stem cell cultures. So mesenchymal stem cells, or MSCs, are multipotent stem cells being researched in the fields of regenerative medicine and tissue engineering to artificially reconstruct damaged human tissue. Unfortunately, a high number of cells are required for research and clinical purposes, and current planar cell cultures produce low yields. So our lab sought to solve this by moving from 2D cultures to scalable 3D porous scaffolds. The benefits of this transition are an increased surface to volume ratio and the scaffolds being more mimetic to biological environments. Despite the promising direction, the cells that were seeded into the scaffolds were not homogeneously distributed. As you can see in the area in yellow, had a high number of cells, but the red area had fewer. Our project focused on overcoming this by changing the scaffold geometry to a more complex shape known as isotres, and plasma treating its surface for greater homogeneous penetration. To achieve our aim, 4 scaffolds were 3D printed by a resin printer. Though the scaffolds contained biomimetic features to improve distribution, the geometry alone wouldn't make the scaffolds compatible with MSCs because the surfaces were hydrophobic. To resolve this, 2 scaffolds were treated using plasma immersion ion implantation. The surface was bombarded with plasma nitrogen ions to make it more hydrophilic. We needed to test the scaffold's water uptake and retention capability, so we placed the scaffolds into a pool of dyed water and observed the distance water traveled through capillary action. We also needed to check for MSC attachment, so a cell attachment assay was performed. Microscopy could then be used to determine the abundance of cells attached. So here you can see the treated and untreated scaffolds being placed into the dyed water prior to cell seeding, and that the geometry combined with plasma treatment increased the water uptake of the treated samples. The treated scaffolds also had one more layer of height than the untreated ones, further indicating the water uptake of the treated scaffolds increased significantly as the dyed water was able to reach the fourth and highest layer and be retained. So these results suggest the plasma treatment successfully increased the hydrophilic nature of the scaffold surface. The treatment combined with the geometry allowed for greater water uptake and retention. While the scaffold hydrophilicity is the key step, minimal quantity of cells were observed under the microscope, which suggests there could be a problem with cell attachment and that bioactive molecules are required. So more research is needed to determine whether the MSCs could be distributed more homogeneously. So the next step is to functionalize the scaffolds with cell adhesive molecules and optimize the surface to maximize the cell yield. Thank you.
Thank you, Daniela. Fantastic, very well done. Question from Mark Lawrence. Do you think your IsoTrust configuration was optimal? What other designs might be better?
Good question. So the initial design was more cubic, and what had happened is the cells kind of concentrated in I guess the first two layers of the scaffold, and so that design came from a Nature, like a research journal, and I think due to the surface area of the design inside the cube, it helped with the water retention. So there could be, I'm sure there's room for experimentation. Yeah.
Endless geometries. Yeah. Okay, well, that's great, Daniela. Thank you very much for that. And our next speaker is Takwa Tusawi. So over to you, Takwa.
Yep, hi. I'll be sharing my screen in a second. Oops. All right. And, my bad.
Brilliant. We can see.
Okay. Can you see that?
Yeah, looks great.
Yep. All right, cool. So hi, everybody. Today I'll be talking about my project, an analysis of investment treaty corporate social responsibility provisions for public health and nutrition. So we know that CSR activities can be an important contributor to public health and nutrition, and the documentary analysis that I undertook over the past 8 weeks shows that more and more countries are including detailed provisions in their international investment agreements to encourage CSR. So a little bit of background. International investment agreements sit at the international level of policy, and they're signed between two parties, which are individual governments or groups of governments, and they apply to investments. So investments include Macca's setting up restaurants overseas, and therefore including the food environment. The problem is that the historical development of these international investment agreements has been imbalanced. It favors investor protection over states' policy objectives at the cost of governments. And so a recent development has been the inclusion of corporate social responsibility provisions to rebalance these investment agreements to achieve state objectives, including public health objectives, especially in the context of sustainable development. So our aim was to examine the ways in which these agreements encourage CSR, align with government priorities for sustainable development, and in particular nutrition. So firstly, what we did is we got a database of agreements from the electronic database of investment treaties and used a keyword search of corporate social responsibility, responsible business conduct, and social responsibility to search for agreements. This generated a data set of 105 agreements with inclusions of corporate social responsibility, excluding model agreements, and 2 investment agreements that were deemed inadequate for the data set. And so what we found is that the percentage of international investment agreements with CSRs signed each year has increased significantly since 2006, as we can see in this graph. And the top 3 parties with the highest number of inclusions were Canada with 21, Brazil with 15, and the EU with 15. Following this, we extracted text from these agreements that had CSR inclusions or public health inclusions or sustainable development inclusions. And then we undertook a documentary analysis of characteristics of the inclusions and trends. This, along with extensive background reading into the history of CSR, led us to develop a level of detail typology to categorize these texts of CSR provisions from low to high. So an example of a low level of detail inclusion would be this corporate social responsibility article from the Australia-Indonesia Bilateral Investment Treaty. And as you can see, it's relatively short, does not name any specific CSR guidelines, and has no description of CSR. On the other hand, this is an example of a high level of detail CSR provision found in the Brazil-Ethiopia Bilateral Investment Treaty. As you can see, it is much longer. It refers to specific guidelines called the OECD Guidelines for Multinational Enterprises and lists 11 dot points that describe what CSR is, what they consider to be CSR, as well as a specific reference to sustainable development. So in summary, we know that CSR can be an important contributor to public health and nutrition. And given the imbalance in international investment agreements, more and more governments are including these kinds of CSR provisions to rebalance these investment agreements and achieve public health objectives. Thank you.
Thank you, Takra. That was beautifully clear. And let's see how we're going on chat. Any questions? I think we're coming to the end of the program. So, but Takra, it was very interesting to see the, I guess, asymmetrical distribution amongst nations in take up of CSR. So Australia doesn't seem to be doing terribly well by all accounts.
No, actually, I didn't include it in this presentation, but I am doing research on the makeup of the makeup of the regions in this data set. And we found that Brazil was definitely a standout. It dominated the medium, high and high categories. The EU as well was very advanced in terms of its focus on sustainable development and the role of CSR in contributing to sustainable development. The good thing though, I think, is that we've seen a variety of countries. So it's not only high income countries or low income countries, it's quite spread out. And that really lends to the potential benefits of CSR for public health and nutrition.
So I guess the key question, Sarah Hill asks this, do we have evidence that CSR works in relation to achieving the SDGs, particularly in relation to nutrition?
Yeah, so there is definitely a fierce debate about this because on one hand, CSR really encourages businesses, especially through stakeholder theory, to take into consideration impact on not just shareholders and on their returns, but all stakeholders involved in the supply chain as well as consumers. But on the other hand, we have concerns that CSR might be misaligned just to benefit the profits of these companies. The idea is that at this stage, we wanted to see that whether countries are interested in making these high level changes to ensure that international businesses, these investors, when they go into the host country, they're abiding by the rules and they're not going against anything that would go against the public health objectives of these countries. So it's definitely something to investigate in more detail.
Great, thank you, Takra. Great answer from, says Sarah, and I agree. Excellent. So now our final speaker, Matthew Yang, is a pre-record. So we've run over a few minutes, but my goodness, given the size and extent of the program, that's pretty remarkable. So if you don't mind, another 3 minutes and a few extra comments from me, then we'll press on. Thanks, Sarah.
Hey guys, my name is Matthew, and today I'll be presenting about how I was able to successfully isolate histones from mice liver tissue samples and the implications intermittent fasting had on any epigenetic regulation. So first of all, what did I do and what is intermittent fasting, and in particular, every other day fasting? Every other day fasting involves alternating days of limited food intake and ad libitum consumption. Ad libitum just means as much as one wishes to eat in this context. And you can just see that in the diagram up here. And next you may ask, what is epigenetics? Well, it is basically when our body makes small temporary modifications to our histone without changing our nucleotide or DNA sequence directly. And as seen in this diagram here, on the left inside our liver cells, we have our DNA in the nucleus, which is condensed by these proteins called histones, which are arranged in an optima seen as each of these lumps of balls here. Thus, in this study, I aim to analyze whether every other day fasting would have any epigenetic implications when analyzing mice liver tissue. And I aim to do this by following and adapting a protocol referenced down here, where they were able to successfully isolate histones from cell cultures for proteomic analysis. So here on the right, you can see a Camilla gel with a pretty consistent set of bands where the green arrows are. And these clear distinct bands at around molecular weights of 15 to 20 kilodaltons is consistent with core histone proteins, while these ones at 30 kilodaltons are consistent with the histone H1 linker proteins. Then these blue arrows just highlight 2 of the fasted tissue samples seem to have contaminants or impurities, as there's quite a bit of noise and smearing with the gel. This could be hypothesized to be due to the fasting intervention causing changes to the structures of nucleosomes, making the fasting samples more prone to contamination. On the left here, this is just a figure showing the propanylation efficiency of a protocol, where all the lysine residues other than the acetylation and dimethylation shown here have been successfully propanylated, which is just these red bars. And this is usually quite hard to achieve successfully. And that is why it is quite amazing that our protocol was able to achieve this and with such high efficiency. Next, I used the data from the mass spec to do an enrichment analysis of what types of genes I have in my samples and what are their functional implications. This network on the left just shows the genes that are interrelated. And in the final sample, all we need to focus on though is the colored dots, which belong to larger families of organelles within the liver cells. The yellow dots represent histones, green spliceosomes, red ribosomes, and blue mitochondria. And from this analysis, it didn't take into account the intensities at all and purely just presents the data based on if a gene is present or not. Thus, I had to do some further quantification to clearly exhibit how successful the histone purification was. And this bubble plot on the right basically is a plot of the sum of the intensities of a random 8 genes from each of these 4 organelles, and it's just the total intensity. And as it can be appreciated, the histone intensity and abundance is much higher than that of the contaminants seen in the diagram on the left, therefore reaffirming that our gel results from before were indeed banned for histones and that we have successfully isolated the histone from our mice liver tissues. Furthermore, on all the histones we purified for each of the liver tissue samples, we were able to use mass spec to look into specific histone modifications. On this graph on the right, this showcases one of the modifications I chose to look at, which is just histone H4 lysine 32 acetylation. There may appear to be a little bit of variability between the fed and the fasted samples, but looking closer at the intensity on the y-axis, it is pretty clear that they are not that different. I also did a student C-test to test for significance amongst any of my histone modifications, not just this one, but all of the p-values were not statistically significant, leading me to perform a power calculation for all of them on RStudio to calculate the theoretical number of samples I would need to compare before there would be a significant difference between the samples. In this particular modification on histone H4, the N required was 26. However, on many of the other mods, the N would be close to 100 or even in the thousands, making it quite insignificant or unfeasible. Overall, despite the fact that there seemed to be a lack of significance between the fasted and fed treatment groups, our protocol opens the avenue for future experiments to utilize mice liver tissues to successfully perform histone epigenetic analysis via mass spec. And they can even use this in a more focused way for other mods such as histone H4 lysine 16 acetylation, which is said to play a role in carbon metabolism and obesity, which is quite relevant to this intermittent fasting study that was done here. I would now just also like to say a big thank you to my supervisors, and in particular, Dr. Lawrence, for all the assistance and guidance they have given me during this research period. And thanks for listening to my presentation.
Well, thank you to Matthew, and thank you to all our speakers. We've just heard 22 talks. We've run over by 10 minutes or thereabouts. Sorry about that, but my goodness, it was worth it. They were, all of you, remarkable and testament, I think, to the value of this summer scholarship program and to the power of collaboration across the Charles Perkins Centre. So well done to everybody, the students, the supervisors, thank you to the audience, and thank you particularly to Sarah Anthony, who has coordinated this as she does each year, and it's getting more and more complicated a task, not made any easier by COVID. And Sarah, thank you. And thank you to Nancy as well. The team have been wonderful. We will, I think, provide certificates in due course, and Helen Laughlin is sure to chase some of you for stories for the media. We'll make the most of the opportunity. So please look forward to that. And thank you, everybody. And well done again. It's really remarkable. Thank you.